Today, in this relatively short 15 minutes talk, I'd like to share some idea and some of our current experimental results on how interaction with environment, especially interaction with freeze and thawing environment, give rise to protocell dynamics and shape protocell individualities. All right, so first, I'd like to start with a big question. How primordial cellular life originated? Of course, this is an extremely big question. It's too big, probably too big to answer directly in a single study. But for us, working in the field of synthetic biology and artificial cell biology, we often break this big question into several key fundamental processes or key features and explore them by constructing such a system in a bottom-up way. So here in this talk, I will break the big question into the three fundamental key processes that is essential for procell to become an evolutionary individuals. So the first topic is about biomolecular system assembly. So without sophisticated and complicated machinery, how did procell assemble our functional systems? The second topic is about procell growth and content mixing dynamics. That is essential for complicating the cellular systems. And third topic is about selections. Once procell acquire distinct dynamics, what we may call distinct individualities, can this difference or can this diversity become a target of selection? So these three topics are what I want to explore in this talk. And this, the next question is about what kind of environment will support all of these three processes. Because when it comes to the process which doesn't have a sophisticated machinery, it's necessary to interact with the environment to drive these processes. So what environment? So here we focus on freezing thawing environment. The freeze-thaw cycles are ubiquitous non-equilibrium processes driven by temperature fluctuation across the freezing point of water. So as long as liquid water and ice coexist, freeze-thaw can naturally occur. For example, on the areas and the icy moons as well. But the point is the process can be continuously exposed to environmental fluctuations here. And also, freeze-thaw provides an effective concentrating mechanism. During freezing, the formation of ice crystal concentrates the biomolecules into liquid brine pockets. This concentrate mechanism is well known, and previous studies have shown the ribosome assembly and DNA hybridization and ribosome concentration in this liquid brine pocket. So if you want to assemble some system, or if you want to promote fusion event, it's some kind of concentration mechanism is required, and the freeze-thaw would be the perfect candidate for this purpose. And also, freeze-thaw induced transient membrane destabilization by ice-induced mechanical stress or the phase transition driven membrane destabilization. And I think this will be the major cause of membrane the content encapsulation or content mixing or fusion. So here we leverage these universal properties of freeze-thaw cycling. We aim to reconstruct key processes of protocell, starting from very simple 100 nanometer vesicles. So first, we examined molecular assembly into the vesicles. We first tried to encapsulate casein, the very simple small molecules, and GFP, this is the protein macromolecules. And what we did is first we prepared a 100 nanometer size vesicle made out of these three types of phospholipid. these guys differ in number and position of double bonds. For example, PRPC has one double bond here, and PRPC has two double bonds here and here, and DOPC has two double bonds, but in the different positions. So what we did is to mix empty best core and Gaussian or GFP and performed free cell cycles. And here in the right panel shows the Gaussian encapsulation and GFP encapsulation. And y-axis is the log scale. And also we try to compare the freeze thaw condition. So what we did is to compare the fast freezing, fast thawing, fast freezing, slow thawing, etc., etc., etc. And what we found is that the slow freezing and slow thawing enhance the encapsulation of both Carlsen and GFP. And also when we compare these three compositions, we found that DOPC encapsulates the most, followed by PLPC and POPC best scores. And why? So we visualize the best score after freeze thaw, slow freeze and thaw cycles, and we found that the DOPC and PLPC best score grow to tens of micrometers, which is likely to enhancing encapsulation capacity compared to POPC. So next, I try to assemble more complicated system. What we used here is pure system, which consists of 69 defined components, including enzyme, ribosome, and some small molecules, which must assemble correctly to function. So basically, what we did is to mix empty vesicle, 100 nanometer size empty vesicle, and pure system, plus GFP encoding DNA. If pure system assemble and the functions GFP is synthesized and emit fluorescence. And look at this, the figure B, we saw an increase in GFP fluorescence in POPC and PLPC and DOPC vesicle, which means the pure system assemble and function inside the vesicles. And here is we also confirm the pure activity under microscopy. So the next, we asked whether freeze thaw could concentrate pure system in diluted environment. So to test this, we use 0.3 times diluted pure system instead of non-diluted pure system and performed pure freeze thaw cycles. In the bulk condition, this pure concentration doesn't work. However, after freezing thawing condition with vesicles, we observed the GFP expression inside the vesicle. And some vesicle shows very strong GFP signals. So this shows that the freeze thaw concentrate and assemble pure system within the vesicles, even at the concentration insufficient to support pure system activities. So to conclude topic number 1, we saw biomolecular system assembly and function inside the vesicles. And at the same time, we saw some vesicle size increase, vesicle growth. So next, we move on to the growth event, including content mixing in details. So specifically, what determine individual growth dynamics under freeze thaw? What we focused is the structure of phospholipid, especially the degree of unsaturation. So as shown in this time image, we found the difference in the growth behavior depending on the lipid composition. So for example, POPC vesicle predominantly grow by aggregation, whereas PLPC vesicle grow mainly through membrane fusions. And also as shown in this figure, the x-axis is degree of unsaturation, the y-axis is a fraction of growth. And we found so the clear trends that more unsaturates the membrane, the higher the growth fraction. So these results indicate that physical-chemical properties coming from lipid unsaturation plays a key role in the growth dynamics. And in the interest of time, I'm not gonna into detail, but when it comes to the content mixing, we also see the clear dependence on lipid composition. So to conclude topic number 2, we found that growth and content mixing dynamics depends on lipid composition. This raises the next key questions. If composition determines protocell individualities, can diversity in composition drive selection? To test this compositional-driven selection, we quantified the changes in lipid composition before and after freeze-thaw cycles. And what we found is that PLPC is selectively enriched in the grown vesicle after freeze-thaw cycle. So we've successfully demonstrated the lipid compositional selections. And further, to examine how internal molecules retained after freeze-thaw, we encapsulate DNA, different DNA, as internal molecular markers. And remarkably, DNA originally encapsulated from PLPC also become preferentially retained after freeze-thaw cycle. So this indicated that the lipid compositional information determines the protocell properties and specific lipid composition, and internal content will be selected after freeze-thaw cycles. And here is the conclusion, and thank you.